Good evening, everyone. Welcome to Hope on the Horizon, a very special live stream webinar we have developed with our friends at Johns Hopkins Medicine. What better way to spend the evening of World Pancreatic Cancer Awareness Day than together? Thank you to our Chief Medical Advisor, Dr. Elizabeth Jaffe, who got the ball rolling for this special event. For the next hour and a half, my colleagues and I will discuss a range of topics that we've chosen with you in mind. As the world's largest private funder of pancreatic cancer research, we believe research is fundamental to giving patients and their families more hope and more time. The Foundation's funded science has been a driving force in every major advancement in pancreatic cancer research. And thanks to you and the scientists whose critical work we support, pancreatic cancer research is now advancing faster than ever before. The Lustgarten Foundation was created to advance the research related to the diagnosis, treatment, cure, and prevention of pancreatic cancer while raising public awareness and providing support for patients and their families. Like many of you who are joining us this evening. And just one brief housekeeping item, we welcome your questions. Please type them in the Q&A box at the bottom of your screen, and we'll do our best to answer them at the end of each session. I'm very pleased to be joined now by Beth Honors from Johns Hopkins Medicine. Beth is a senior GI immunology oncology research nurse at Johns Hopkins University School of Medicine and my co-host tonight. Welcome, Beth.
Thank you, Linda. I'm very happy to be here tonight with everyone as we talk about our research being done at Johns Hopkins for the fight against pancreatic cancer. Johns Hopkins is a world leader in the diagnosis and management of pancreatic cancer, leading all other institutions in novel therapies, innovative clinical trials, and in training the next generation of pancreatic cancer experts. It is also very timely to have this webinar in the month of November, which is Pancreatic Cancer Awareness Month. So welcome to everyone who was able to join in tonight.
Thanks so much, Beth. Now, I'm pleased to introduce the newest addition to the Lustgarten research team, Dr. Se-Jin Chung. Sage is our program scientist and joined us earlier this year, having done her postdoc at the Salk Institute in La Jolla, California, one of Lustgarten's 6 dedicated labs. Welcome, Sage.
Hi, Linda. Thank you for having me.
So Sage, you're going to kick things off by explaining how clinical trials begin with Lustgarten and what is important about our focus in the earliest stages of development.
So let me start by sharing my screen. Okay. Awesome. So thank you again for allowing me to talk. I'm really excited to not only talk to you guys about clinical trials and vaccines, but also to hear from the nurses at Johns Hopkins as well. So to begin talking about clinical trials, I just want to give this very simplified overview of the clinical trial stages. Essentially, in the preclinical stages, we have investigators that are conducting experiments to really understand their drug of interest or combination therapies in their disease of interest. And they perform experiments to really define the mechanism of action or find the effect that they believe is worthy of a clinical trial and onto the clinic. Once those studies are finished with the appropriate approval, the clinical trial stages begin with usually the phase 1 studies where really it's focused on a small group of people and to really understand if there are any safety issues of the dosing or any side effects that cannot be seen in the animal models or in the cell culture. As the stages progress, it moves on to a wider range of people, up to about or hundreds of patient population in the phase 2 studies. And in this phase, it's really focused on the efficacy of the drug or the combination of interest. Of course, with the safety still being in mind, but because phase 1 has already been completed and approved, phase 2 really looks at, is the drug or combination effective, and are we seeing what we want to see? Then in the phase 3 trials, at this point, the drugs were proven to be efficacious, and it expands into a larger cohort of patients and a little bit more diverse and broadened out to thousands of patients to really see the effect in a larger audience of patients. And once that is all complete, the FDA will review and approve it for the clinic. While it goes into the clinic, there are still studies that could be conducted to really understand the long-term effects of the combination or the therapy in question. So really, when you think about the clinical trial stages of how a drug or a therapy goes into the clinic and into the doctor's office, it's really a long and arduous process, time-consuming, and also costs a lot of money to have the appropriate personnel and studies that are done. So at Lustgarten, we really wanted to ask, how can we make the most impact in this phase, in these stages, because we really want to get patients better care and better therapies. For us, in the pathway to the clinical trial that we found to be most impactful was essentially the transition from preclinical to the clinical trials. So this again consists of the investigators conducting their discovery and development studies to really identify the drug of interest or the combination of therapies in the disease, and then conducting the preclinical research studies that are appropriate for clinical trial approval. And once these are done, it goes into a protocol submission into the IND, and the IRB, the Institutional Review Board, will look at the protocol and approve it for the appropriate phases. So this is where the Clinical Accelerator Initiative program comes in from the Lustgarten, because we believe that this was the area that had the highest activation energy and the most cost-related area that we could really make an impact on to speed up the process. And the goal of this program is to support the clinical testing of the novel therapies or the combination approaches to advance the translation of the new discoveries and really increase the number of pancreatic cancer clinical trials that are available for the patients. So when we think about preclinical, this can be a large range of studies, and this is just a small subset of topics that we are looking at when we think about the preclinical studies that are ready for clinical trials. So in phases in the resectable tumor, as well as metastatic setting, and really covering immunotherapy combinations, as well as chemotherapy in the neoadjuvant, as well as the new and exciting vaccine therapies that we'll be talking about later. And once we are able to support the investigators in advancing the preclinical studies to the clinical trials, we really want to continue facilitation of the collaboration across the studies to further increase the trials and continuously improve the trials for the patients. We also encourage data sharing to, again, continuously improve the trials because we know that things can happen, and we really want to be able to have researchers talk to each other and make sure that the patients are getting the right care. We also know that having data available as scientists provides the foundation to pave the way for a new discovery. And so we really want to be able to support that region as well. So as part of the Clinical Accelerator Initiative, we have a translational advisory group, which consists of experts to implement this program. It's led by the Chief Medical Advisor, Dr. Elizabeth Jaffe of Johns Hopkins. And this group is really there to identify and review any of the potential pancreatic cancer clinical trial concepts. The group meets at kind of a monthly basis, but really any time that there is an investigator that has something that they want to present for a clinical trial, and we solicit proposals from academic institutions, foundations, as well as contract research organizations. And so for this clinical accelerator initiative, it's really important for us to know that we are helping in this early stage for the investigators, because we know it's really important and we know that clinical trials take a very long time. But we also know the importance of doing these trials at a quicker rate so that we can find the therapies for the patients and work together in finding a better treatment. And so with that, I'll take any questions.
Sage, I see a sorry about that.
That's okay.
I see a question in the Q&A box about the
Okay.
CD40 agonist antibodies and the PD-1 inhibitor with the TIGIT.
Mhm.
And I know that's one of the studies and the research that we're really proud of, and the person wants to know about where that might be opening.
Yeah, so it's actually open, and I can actually, so can I type the answer, because I have the clinical
Yes.
trial number, so
Great.
I can
So you
do
can go ahead
that,
and
and
answer
that'll
that one.
have that'll have the appropriate contact. So I can just go ahead and do that.
Okay, terrific. So that was the only question, so we can
Okay.
move on to our next segment. Thank you, Sage, for laying the groundwork for the next part of our conversation. So Trish Brothers and Beth Honors will talk about what clinical trials are underway at Johns Hopkins and how you might be able to participate. I talked about Beth's amazing credentials before, and Trish is just as fantastic. Trish is the clinical trials referral nurse coordinator, a very big job, and senior research nurse for GI immunology cancer research trials at Johns Hopkins. So welcome to you both. I'm really excited, and I'm sure our audience is really excited to learn about what trials are underway at Johns Hopkins. Welcome.
Thank you, Linda, and I'm really excited to be here. I'm going to talk about the clinical trials in that we have open at Hopkins. Right now, we always have different trials opening up. There's always a lot of background work that goes on at Johns Hopkins and at any cancer research institution before they actually are able to enroll patients. I'll talk to you about the ones that are actually able to be enrolled. So we have clinical trials for all different stages of pancreas cancer. We have trials for neoadjuvant, and this simply means before surgery. These are for patients that have been unfortunately diagnosed with pancreas cancer that has just been confined to the pancreas. It's nowhere else in their body but just in the pancreas. So that we have trials in that space, and then we have trials in the adjuvant space, which is after those patients go to surgery. So the neoadjuvant portion is where they have cancer, pancreas cancer that has started just in their pancreas. They will get trial treatment, and then they'll go to their Whipple surgery. And then they have another portion of adjuvant treatment, which is after they have the Whipple surgery. And this is done to prevent any type of spread, any cells, cancer cells outside of the pancreas that may want to make tumors in the future. Giving drugs at that time after the surgery prevents that from happening. That's the hope. So our neoadjuvant and adjuvant trials are combined at Hopkins. So we always give a pre-surgery treatment, a surgery, and then an after surgery treatment. So our first two trials that we have open in this space, one is made up of vaccine and another immunotherapy boosting agent. The second one is immunotherapy plus oral targeted drugs. And what a targeted drug is that it just cuts off pathways to the tumor if the tumor expresses a certain target. Then we have trials for locally advanced or unresectable. That's for pancreas cancer that has been diagnosed, but it's just in the area of the pancreas or in the very near surrounding areas, not widely spread to far away parts of the body. So we have one trial in that space that's open right now, and the patients would be screened for their trial, then they will go, if they aren't local to Johns Hopkins area in Baltimore, they would go to their local oncologist and get chemotherapy, one of the standard first-line therapies that we give for patients is FOLFIRINOX. Then they'll come back to Hopkins. They will get a scan to make sure that it's still, the pancreas cancer hasn't spread and it's still in the area of where it started. And then they'll get the study medicine. They will also get some targeted radiation therapy. Then they'll go to their surgery. And then they'll get the adjuvant portion of the study drugs after they get the surgery and they heal from the surgery. And this includes the chemotherapy, that's standard of care, with immunotherapies and the plus or minus the cancer GVAX vaccine. Then we have trials for patients that are newly diagnosed and the pancreas cancer has started not just in their pancreas, but has also spread to faraway sites like their lungs or their skin or their liver. We have several trials in this space for patients who haven't been treated and have just been newly diagnosed at this stage. So we have one that is, it's a randomized trial for chemo with an immunotherapy boosting drug, and then another oral agent that also boosts the immune system. Most of our trials in this space will have one arm where it is the standard chemo that we know works for some patients, and we're adding extra drugs on different arms to see if adding those extra drugs can actually give a better benefit against cancer. That's what we're being tested. And then we have another trial that patients are randomized to the standard of care chemotherapies that's widely prescribed in the communities and outside of a trial, gemcitabine or nab-paclitaxel, or the FOLFIRINOX cancer treatment regimen. And what's interesting about this trial is that they actually take a pre-treatment biopsy, and the biopsy is actually goes to 6 different labs around the country and in Canada. And we have researchers developing organoids based on the tumor itself of the patient, and it will guide the next treatment option should the patient need another treatment after this first-line treatment. As we know with pancreatic cancer, we always have to think 2 and 3 steps ahead. Another trial that we have in this space is just a low-dose chemotherapy regimen. The thought process is giving 5 different low-dose chemos will hit the cancer cells at different times of their cell cycle replication to kill more of them. And then after 6 months of treatment, the patient will go on to receive dual immunotherapy plus one of those oral pills. And then we have a cancer vaccine opening in this space for KRAS mutated pancreatic cancer that hasn't been treated. It's a vaccine plus immunotherapy, and that will be opening in spring of next year. And then we also have trials for patients who have received other chemotherapy, other treatments for their pancreas cancer that has unfortunately spread outside of the area of the pancreas. We have one that has it's combination immunotherapy treatment. So it's immunotherapy plus an immune boosting drug. And immunotherapy works differently than chemotherapy, where it stimulates your own immune system to recognize the cancer as something that needs to be attacked and attack it. And then we have a 4-drug combo that has something that agitate the immune system, an immunotherapy, and then a pill immune booster. We also have a trial for a very specific type of pancreas cancer that has spread outside of the pancreas, and it is for adenosquamous, and that is something that you see in the tumor itself. And I'm not a scientist, but it's under the cell, under the microscope, the cells look different than the very common type of pancreas cancer, which is pancreatic adenocarcinoma. So this is adenosquamous, and we have one there with immunotherapy agents as well. And I would like Beth to join me to see if she has any questions or if she wants to add anything, because I know that she is one of our research nurses who have been giving many of these drugs for many patients for many years.
Thank you, Trish. Yes, just one thing to say, when I first started working in immunotherapy with Dr. Jaffe about 21 years ago, we only had one study, and that was for resected pancreas cancer. And now, as Trish just showed everyone, we have it for neoadjuvant, adjuvant, locally advanced, metastatic. So there is a lot going on at Johns Hopkins. And we're just working very hard to hopefully find, a treatment for pancreas cancer. So there is hope out there that we're using immunotherapy and vaccines in many different ways. So thank you, Trish.
You're welcome. So for more information about any of the trials that I just talked about for pancreas cancer at Johns Hopkins, this is the email address and the phone number for the GI Clinical Trials Referral Office, which I am a member of that office. And we'll be happy to look up your information and see if one of these trials would be a fit for you or your loved one. And then, excuse me, if there is another trials that you want to look at for pancreas cancer, there are 2 websites to check out. Let's Win Pancreatic Cancer helps with trials, looking for trials across the country, as well as clinicaltrials.gov, which shares where you can search pancreas and what area you live in, and we'll have all the trials that are open with the contact information of those study teams to get in touch with them to see if you or your loved one is a match for one of their trials.
That's great. Trish and Beth, thank you so much. I just want to remind everyone who's joining us this evening that there's a lot of information here, and there may be terms you don't understand. If there's something that someone mentions you don't understand, please feel free to ask a question using the Q&A box down at the bottom of your screen, and we'll be answering your questions either in person using the chat box, or if relevant to the entire community, we'll answer that live. But I'm going to briefly interrupt our agenda to ask our program scientist, Dr. Chung, to talk a little bit about what an organoid is, because Trish mentioned an organoid, and that might not be something that everyone is familiar with, but is certainly of very much importance to the Lustgarten Foundation, to Johns Hopkins, to the entire PASS-1 trial, which is so important. So Sage, take it away.
Yeah, I'm glad you asked me. I typed up the answer, but I realized that probably not everyone can see it. So an organoid is essentially a mini version of the organ in question. So in this case, it's the pancreas. And this is different than what we would normally use in the lab with just regular cancer cells because they would just grow flat. So they're two-dimensional, and that really determines, or that can differentiate the way that they respond to a drug, rather than growing in a spherical manner that really recapitulates probably the structure of the organ in a better way. So the organoid growth is, these cells are grown in an embedded material. We call it matrigel, and it's essentially structures or provides the nutrients and as well as a structure for these cells to really shape into a spherical structure. In the example of the pancreas, the organoids actually do look like just a sphere. And these are really critical for really understanding more so about how the drug can access the tumor, as well as, you know, how the tumor grows. And these have been increasingly gaining popularity, not only with the Tuvison lab. There's been a lot of labs that are studying it, not only in pancreas cancer, but also in the tumor microenvironment space as well, because with the organoids that are growing as the miniature organs, you can really add in different components of the microenvironment as well, and really understand how they can interact with each other.
Thanks, Sage. And we're really very hopeful that precision medicine will be the future of where we take pancreatic cancer chemotherapy, because right now the two standards of care, which we know are FOLFIRINOX or gemcitabine Abraxane, we know that different patients react differently to those two standards of care, but we don't know why. And we're hoping that as we move through the PASS-1 trial, that some of those questions will be answered. And also eventually we hope that the organoids can then be used eventually for the next therapy. And maybe even someday we hope that they could even be predictive in the neoadjuvant setting. So in the adjuvant setting. So there's so much that we want to do and need to do. And we're so grateful to all of you out there who are participating in trials or are thinking about participating in trials. You're the reason why we're able to keep pushing all of this research forward. There's two questions here that Sage, you might be able to answer. For the squamous versus the adenocarcinoma, do you need a biopsy, or does a biopsy of the liver tell you which of these you have? And I'm not sure if you can answer that.
We
might
don't
need a
feel
physician
very confident
we might need a
of saying that,
physician
but
to do that.
I do believe that you would need the biopsy of the pancreas, but I don't want to say with full confidence because I haven't actually studied a lot of the squamous.
Okay, and then so the immunotherapies and vaccine study, I didn't see it listed, so we will this presentation will be available to everyone afterwards, so you'll be able to see everything that's available. And so you can see where on the Johns Hopkins site or the clinicaltrials.gov that you'll be able to get that information. And then back to you, Sage, with regard to the TIGIT inhibitor trial. I believe it is only at Dana-Farber. Is that correct?
Yeah, that's what I believe as well.
Yeah,
but
I believe so as well, that that's only being offered at
yeah.
So Trish and Beth, I don't know if you had any more that you wanted to add there before we move on to Dea.
With the adenosquamous, I just wanted to say that I believe it is the pancreas because I've worked behind the scenes with some of our researchers. And oftentimes if they don't see it with the original pathology report or tumor report, we have a dedicated researcher here that will take another look at the sample.
Okay, thank you.
Concerned about that.
Thank you. And this is actually a really, really good question that I see. And so we'll take this one last question and then move on to our next section. And this really gets to the heart of pancreatic cancer, which is pancreatic cancer as compared to other types of cancer, such as lung and melanoma, is more resistant to immunotherapy. And to what extent is this the case? And if so, why? So Sage, do you want to take that? And then I don't know, Beth or Trish or Dea, if you want to pipe in.
Yeah. So pancreatic cancer is notoriously known as what we call a cold tumor. And so what that means is that the immune components, either because they can't access the tumor site very well because of the heavy stroma that surrounds the pancreas, or because of other immune suppressing factors that the pancreatic cancer cells are secreting out into the microenvironment that prevents the immune cells from really doing their job. And so that's why a lot of the single agent checkpoint inhibitors or immunotherapy has failed or have been unsuccessful to the extent that they want to be, because as a single agent, the microenvironment just appears to be too immunosuppressive. And that's why these new trials and the vaccine studies and the combination studies are important, because what they're really trying to do is activate the immune system to really get the cells into get the immune cells into the tumor to really attack the cancer cells.
That was perfect. Thanks, Sage. Beth, Trish, anyone else want to jump in here?
I know with some of the work that we've done with the vaccine and immunotherapy that we do have the patients undergo biopsies while on the study, and they'll usually have a baseline biopsy, and then at some point during the study they'll have a second one. And what we're looking at are what cells are in the tumor microenvironment at baseline, and then after some immunotherapy and doing tests and staining to see. It's great that they're there in the tumor environment, but what are they doing there? Are they active or are they just passive? So it is something we are looking at as well in all of our research studies.
Thanks, Beth. Trish, anything you wanted to add there?
No, I think they both said it all. Yeah.
Great. Okay, just wanted to make sure. And then again, one more question, and this is actually this person kind of gets to the heart of where we would like to see the research going at some point. And the question is, are there clinical trials for patients post-modified Whipple and NED? And I'm going to go out on a limb and say there actually is not. And it's something that we would like to do at this point. We know that there is a huge need for patients who have had surgery, they've done their chemotherapy, they've even possibly had some ablative radiation, and now they're NED, but now what? So we know that that's a huge unmet need, but I'm going to go out on a limb, and Sage can hopefully concur, and I'm going to say that there are no current clinical trials for that.
Yeah, you said it perfectly. Yeah.
Concur. Okay, good. Okay. So we're going to move on to our next section, which is genetics and genomics. And our next presenter is Dea Cunningham. Dea is the senior research nurse and lead for the Pancreas Precision Medicine Program Development. Thank you, Dea, for joining us.
Oh.
Dea, I get asked all the time, what is the difference between genetics and genomics, and why is this so important for patients to know? And we know that precision medicine plays a big part in all of this, and we're looking forward to your presentation.
Oh, thank you so much for a kind introduction. That's correct. There's a lot that precision medicine tries to answer, and some of the questions that our patients already asked, that's one of the aspects and efforts that precision medicine tries to answer. Why are things as they are? So precision medicine, for my portion of presentation, I will talk about precision medicine very briefly, and then we'll talk about genetic testing in more detail. So I'm sharing my screen. I hope you can see my screen. Precision medicine is a new way of looking at disease. And some elements of precision medicine are being implemented in care of patients with chronic diseases like heart disease, Alzheimer's, asthma. But during this presentation, we'll focus on patients with pancreas disease. what precision medicine does, among other things, is to bring focus on genes and genetics and how those elements play a role in health and disease, and why patients respond to treatment in this way or the other. So unlike my colleagues that spoke earlier, everything I talked to you about is patient care, not research. So when we talk about genes and genetic testing, I would just like to mention that there is a law that protects genetic information. Some of our patients most of you patients that are in the audience are familiar with HIPAA. HIPAA is a law that protects patient information in general. GINA is another abbreviation for you to remember, but it's a law that protects genetic information. Before we start talking about genes and genetics, genetic and genomic topics are pretty complex. So I will use genetic testing as a simplified version of explaining genetics and genomics. So before we talk about genetic testing, let's imagine genes being sorted into separate groups. So group number one would be genes that carry hereditary material in family from one generation to the next. This test is often called germline or germline genetic test or just genetic test. Second group of genes are found in tissue sample only, tissue sample like surgical sample or biopsy sample. And because they're unique to that tissue sample only, this test is often called tumor profile. Other names for this test is somatic or genomic. You might have experienced, for some patients might, some may not, but healthcare providers tend to use other names to describe these tests. But if there's anything I would like you to remember from this presentation is this differentiation between two groups of genes, and for that reason we have two different types of tests. So when we request a germline genetic test, we check if patients are born with fully operational genes, or there's some kind of change or mutation that's important for providers to know. One gene that rings bell for most of our patients and family members is so-called BRCA gene. What's required for this test in most cases is saliva or blood sample. But in our clinics, we request this test on saliva sample. So the way we process this is that patients receive saliva collection kit in mail. So supplies are delivered to your home address. Inside the kit is a saliva collection tube. So that's kind of homework assignment for patients to provide a sample and send it back to the lab for analysis. Somatic genetic test or tumor profile analyzes tissue from a procedure like surgery or biopsy. Pancreas biopsy samples are most often than not insufficient for this type of analysis. So if that's the case, we complete this test on a blood sample. There's a lot of technical requirements and decision-making that goes into this process, but in our clinic, I'm designated a person to guide patients through genetic workup. On this slide are some of the commercial labs that are often used for genetic tests. At the bottom of the slide, you have Invitae, Ambry, and Myriad. Those are most often used for germline genetic testing. And then we have Caris, Tempus, Foundation Medicine, and Johns Hopkins labs that are often used for genomic tumor profile tests. We're not endorsing any specific lab, it's just a few most commonly used. So why are these genetic tests becoming increasingly more important? So once we have a diagnosis, provider that's in charge of treatment and care decides course of action and in what order are these treatment modalities applied. With genetic tests, we look for some kind of weak link in those genetic components that could potentially open door for another option in treatment. Just keep in mind that some of our genetic tests do have the limitations, so we don't always find what we're looking for. Another test that's more recently being implemented into our clinic flow is genetic test that's used for long-term monitoring. And this test is most applicable for patients after surgery or in remission. And blood samples are drawn at regular intervals, approximately every 6 months. And we monitor for tumor DNA in blood. So precision medicine is ever evolving field. There's a lot of questions that need to be answered. Apart from genes and genetics, precision medicine embodies also huge research efforts. And one of the big components of it is data collection, data that's overwhelming to humans and machines. But in order to draw big conclusions, we need large amount of data. So we do appreciate everybody's participation in research. If you would like to learn more about pancreas precision medicine at Johns Hopkins, please visit our website. You'll find some important information and patient resources. And I'm happy to answer any questions you may have.
I don't see any questions, Daya. That was really terrific. So thank you for your thoughtful presentation. And now I think I'm going to turn it over to Sage and to Beth to take us into cancer vaccines, which is really the, that's where we hope we're going to go. This is really the precision medicine. How can we help the right patient at the right time with the right medication, and how can we really look at prevention, intervention, both adjuvant and neoadjuvant, and Sage and Beth are going to explain all of that to you. Take it away.
Awesome. Thanks, Linda. So Beth, we're, again, as mentioned by Linda, we're finishing up the wonderful discussion tonight by covering one of the most talked about advances in pancreatic cancer prevention, which is, like we have already talked about, the vaccine. So I'll provide the overview, including some of the research that Lustgarten is invested in, and then will you share the efforts underway at Johns Hopkins after my presentation?
Yes. Thank
Okay.
you.
Awesome. So I'll go ahead and share my screen. Okay. And start. Okay. So again, cancer vaccines. When we talk about vaccines, we know about them with the flu and of course COVID, but why cancer vaccines now? So vaccines can increase the cancer killing potential of T cells. So these are the immune cells in the body, by helping them recognize the cancer cells. And they can also increase the number of T cells to kill these malignant cells as well. So this is really important in pancreatic cancer, as we had mentioned before in the Q&A session, where since pancreatic cancer is such a immune suppressive tumor and it's a cold environment for the immune cells, it's really critical that these vaccines are able to activate the immune system to kill the pancreatic cancer cells. And why now and not before? Well, a lot of the technological advances have occurred to allow for this opportunity. A lot of technologies have allowed for the prediction of new targets, so the neoantigens from the patient biopsies. So those are more personalized vaccines that can be tailored to each of the patient per their biopsy. There's also an improved technology for the delivery systems for the vaccines themselves. Before, in the past, it's just always been a difficulty to get these vaccines into the body and in an efficient and effective manner. And also with the sequencing technologies that have improved, there have been continuous improvement in the biomarker discovery phase. And so all of these kind of combined together to really provide this unique time in cancer immune therapy for the pathway to the pancreatic cancer vaccines. And again, just to reiterate a little bit of the points that I had prior about the usefulness of vaccines in pancreatic cancer, beyond the fact that pancreatic cancer is such a cold disease tumor, there's also a large window of opportunity that is there from the initial mutation of the pancreas to its progression to pancreatic cancer. And so because of this large time frame, there's a really unique opportunity for these preventative vaccines to take effect, especially in the high-risk groups who can really benefit with early interception. And so the high-risk groups involve patients who have family members who have had the disease, or they have a certain mutation that is also evident with the addition of the family members that have the pancreatic cancer diagnosis as well. Also, more than 90% of the pre-pancreatic cancer lesions have KRAS mutations. So this allows for a vaccine that will be essentially off the shelf, so it doesn't have to wait for the patient biopsies to get to the sequencing and to the neoantigen findings to develop the vaccine. We can have KRAS vaccines that are readily available to have the preventative measure, as well as preventing the recurrence from happening. And in the current state of the pancreatic cancer vaccines that Lustgarten is invested in, there's again the primary interception that really tackles on the high-risk individuals. So again, it's those individuals who have a family history of pancreatic cancer or the germline mutation. So this vaccine therapy will then prevent the cancer from initially occurring. There's also the secondary interception, where patients have been diagnosed with pancreatic cancer and they have gone the surgery and the appropriate treatment. So they have the resectable tumor, and they will have they get the personalized vaccine again from the biopsy and the development of the neoantigen vaccine or the KRAS vaccine to reduce the recurrence of the cancer from occurring, because we know that pancreatic cancer has a large incidence of metastatic disease that really takes the patient even after the surgery is completed. And so we're really excited about not only the current state of the pancreatic cancer vaccines that are happening right now, but in the future, we're really looking forward to biomarker discovery that really allows us to predict the responses in the precancerous stage from these vaccine trials that are currently going on. We're hoping for increased screening to capture more patients at high risk. So maybe those beyond the family disease, as well as prediction signatures from the cancer vaccine trials that will allow us to stratify the patients in the future studies, because we know we can better predict the way that they will respond. We also are hoping for an expanded use to the broader patient population. So whether that means in terms of manufacturing scale or in terms of increasing the patient population themselves, this is what we're really hoping for. And lastly, to optimize the current vaccine strategies that are happening right now, because even though it is very exciting and the initial results are promising, we know that there is still a ways ahead in terms of really harnessing the true potential of the pancreatic cancer vaccines to have the full effect. And so with that, I can go on to the questions.
So I think Beth is up, but Sage, there are a couple of questions in the Q&A.
Oh yeah, I
I know.
noticed those popping up, but I can wait until Beth is
Beth is
done.
done?
Yeah.
Okay, that'd be great. Beth, you ready?
Okay.
Okay, let me
Great.
share my screen. Okay. Can everybody see that okay?
Yep.
All right. I'm kind of a novice with PowerPoint myself, so.
Beth, I'm sorry, Beth, is there any way that you can get that into presentation mode? So go to slideshow and hit present.
You can also go to the lower right. There's like the 4 icons. So if you on the right, bottom right, yeah. So the one that has the projector, the next one on the right, last one on the right. That one.
Okay,
Yeah. There you
great.
go.
Thank you very much.
Yep.
It's a team effort. All right, so I'm going to take a few minutes now to discuss the evolution of vaccine studies here at Johns Hopkins. So back in the mid 1990s, immunotherapy was thought to be a novel therapeutic approach to recruit and activate tumor-specific T cells to induce a cytotoxic response. So basically, in simpler words, it's using a person's own immune system to recognize and then attack the cancer. So from surgical specimens obtained at Johns Hopkins by Dr. Jaffe, she was able to maintain and replicate 2 pancreatic cancer cell lines, which encompass the most common mutations in pancreas cancer. So this was the beginning of using an allogeneic pancreatic cancer cell to fight cancer. So in this here, we have the tumor cell in green, and then these cancer cells were then genetically modified to produce a gene called GM-CSF, which attracts certain immune cells, such as macrophages and dendritic cells, to create an immune response. So these cells were then irradiated so not to allow them to cause cancer when given to a person, but still maintaining the mutations. So here's the GM-CSF once given to the patient via the GVAX vaccine, attracts immune cells to the site. The idea is that the patient's pancreas cancer doesn't have all the mutations on their cancer cells as the GVAX vaccine, but they have more of the common mutations. So when giving the GVAX to a patient, their immune system sees these pancreas cells as foreign, creates antibodies towards them, with the goal then that the patient's own cancer cells have similar mutations. So therefore, if the immune system thinks the GVAX vaccine is foreign, then the patient's tumor is foreign, and then the immune system can respond to it and attack it. So some of the conclusions that came out of this first phase one study was that mesothelin is overexpressed in pancreas cancers and very low expression in normal cells. So that is important, and we'll talk more about that in a bit. GVAX vaccine expressed mesothelin, which induced a T cell response. And then the mesothelin as a relevant tumor antigen to further study the GVAX generated immune response targeting towards pancreas cancer. So this discovery led to a phase 2 study of using GVAX in resected patients in sequence with chemotherapy and radiation, similar to what Trish had mentioned before. We use it neoadjuvant and adjuvant in combination with the chemo and radiation. The results from these phase 2 studies were encouraging. However, not all patients responded. So this led Dr. Jaffe and her team to further explore how to make pancreas cancer more responsive to GVAX for resected, borderline, and locally advanced patients. So we are now combining GVAX with various checkpoint inhibitors, such as a PD-1 inhibitor, such as Keytruda or pembrolizumab, PD-L1 inhibitor, such as atezolizumab, and CTLA-4 monoclonal antibodies, and you might have heard them as stated as Opdivo in the news. So while first focusing on neoadjuvant and adjuvant use of GVAX, Dr. Jaffe also felt there was a need to test GVAX in the metastatic or stage 4 patient population. So the results using this combination were not shown to provide an increase in overall survival. However, it did show that the combination of GVAX and immunotherapy was better tolerated and provided a better quality of life. So it was felt that we needed a better way to deliver these vaccines rather than using the GVAX cell lines. So this precipitated another type of vaccine being used, and that's using bacteria as the vector. So it's been well studied that the Listeria monocytogene bacteria presents itself very well to the immune system through the liver. Again, taking the results from the phase 1 study in the 1990s, remember that mesothelin was overexpressed in most pancreas cancer, and Dr. Jaffe and Dr. Lee from Johns Hopkins came up with the idea of genetically modifying the Listeria monocytogenes to produce the mesothelin and combining it with other immunotherapy agents to manipulate the immune system from different angles to recognize pancreatic cancer. So in other words, it's using different checkpoint inhibitors at different stages of or different areas of the immune system to again mount a bigger T cell cytotoxic effect against the cancer. So there are several studies currently underway at Johns Hopkins using this. And then just to reiterate what Sage mentioned earlier with the other vaccine studies, we do have some in development using a messenger RNA platform and a KRAS peptide vaccine platform for pancreatic cancer. So these studies are in development or almost meeting their accrual as well. So Dr. Zaidi's studies here at Johns Hopkins is using a pooled mutant KRAS targeted peptide vaccine in combination with immunotherapy agents such as nivolumab and ipilimumab to prevent recurrence of pancreas cancer. And then this same pooled mutant KRAS peptide vaccine is being given alone for patients who are at high risk of developing pancreatic cancer. This is based on maybe their genetic history and their genetics and their family history, sorry. And to be potentially eligible for this study, the person does have to have a radiographic abnormality in the pancreas that they could follow throughout the study. So both studies don't have any efficacy data as of date. So again, by combining the knowledge of our pancreatic experts here at Johns Hopkins and the Lustgarten Foundation's unwavering support and funding of studies, there is hope on the horizon to develop a more tolerable treatment and ultimately a cure for pancreas cancer. So right now, I would just like to leave with a quote from Dr. Jaffe that she says, vaccines have been used to prevent many infectious diseases. It only makes sense now to harness this power for cancer. And we may now have the technology and capability to make this happen. That concludes my talk. If you have any questions.
Thank you, Beth. That was very illuminating and very insightful, and really covered the bases in terms of where we are with vaccines. There are a couple of questions here, and I think I can probably condense them. But in terms of, is the pancreatic cancer vaccine available now? The answer is only in a clinical trial in terms of a vaccine. And is the vaccine likely to be useful for post-Whipple, post-chemo survivors to prevent a recurrence? We talked about this a little bit earlier. At this point right now, the answer to that is no. Would we like to see a future where that is the case? Absolutely, because we know that that is a high need area. So stay tuned for future trials, future discussions, and the future where we can actually have a vaccine for patients such as that. And we are ending a little bit early. So what I would like to do is have all of the presenters on the screen. So Beth, Trish, Daya, Sage. And since this is World Pancreatic Cancer Awareness Day, I think it would be really great. And Beth, you kind of started us off, why should we be hopeful now? And what is it that gives you hope? So I hope you don't mind. Daya, I'm going to call on you that on World Pancreatic Cancer Awareness Day, what gives you hope?
All
Well,
right.
Do
Go
you
ahead, Beth.
Okay. So I think just what gives me hope is, as I mentioned earlier, I started working in this field back in 2001, and I've seen so many advances in the treatment of pancreas cancer through chemotherapy, immunotherapy, vaccines, even surgical and radiation advances as well. And I still have patients from my first studies who are still disease-free. Now, they were resected and they went on for chemotherapy and radiation. So they did get their full treatment, but it's just very heartwarming to me to know that we were able to help some patients. And I just see it exploding in the future as we learn more and more about immunotherapy and the best way to deliver them to make the pancreatic cancer more of a hot tumor, as Sage mentioned, not so much a cold tumor, and make it more responsive to treatment.
Thanks, Beth. And our friends at Johns Hopkins are really on the front line of working with the preeminent expert in immunotherapy, the Lustgarten Foundation's Chief Medical Advisor, Liz Jaffe. So they really know what is next and where we're going, and why there is so much reason to be hopeful for patients, and obviously not tomorrow, but certainly hoping in the coming years. So Daya, if you would take us next.
Yes, thank you so much, Linda. Yes, I'm for that reason, I'm very excited to be part of precision medicine effort, because with the recent developments in science and technology, even in very recent times, you see a big progress. So some of the tests that I talked about during my presentation We did not even have routinely in our clinics prior to 2018. And genetic testing panels expand. We learn about new genes. Those through research, those genes are being incorporated in patient care. So I feel that it's different being patient today than it was 5 or 10 years ago. So like I said, I'm really happy to be part of this precision medicine effort.
Thanks, Dea. Trish, over to you.
When I get new calls for a trial, I always, hear patients and family members. I can hear the fear in their voice, but like Beth said, I've been at Hopkins not as long as her, but for 11 years, and I started as an infusion nurse, and I treated some of the pancreatic cancer patients in the infusion space. And before we had new innovative treatments and trials, I wouldn't be seeing those same patients today, and some of them I still see. And so that's really hopeful, and that's why I know it's one of the reasons why I keep doing what I do to try and offer those treatments for more patients to combat this disease.
Thanks, Trish. We hear so much about what's happening in the medical community with COVID and how difficult it is to staff the trials. And we know that that's difficult. And what I've been so impressed by the resilience of the pancreatic cancer community and the research community, very, overall, in terms of they just want to help patients. They want to provide more treatment options. So I just want to kudos to Beth and Trish and Dea for their commitment to patients and for really wanting to see this through to ensuring that we are getting those better treatment options for patients. And I'm sure that some of you are on the front lines of the COVID pandemic. So we do want to thank you for all that you did to ensure that patients were still getting treated during the pandemic. So Sage, over to you.
Yeah, I don't want to repeat everything that everyone said, but I think the main theme is that there's just been a lot of progress that's been made so quickly. I studied pancreatic cancer since grad school, so 2015, and then organoids I didn't even know what organoids were. Immunotherapy was also very new, and even at the end, so towards the end of my graduate career was when these things just started coming up, and I felt like I was just behind even as a pancreatic cancer researcher. So the fact that everyone had put so much effort and in such a quick and efficient manner to put all of their efforts into finding new ways to model pancreatic cancer and to understand the disease, I think that gives me a lot of hope. And again, as a researcher in the lab, I didn't really get that much interaction with patients or clinicians. And I think through my interactions with investigators in Lustgarten, I think has just opened, even more excitement, because these clinicians are the ones that are seeing the patients, and they're so dedicated and just so willing to, have their team tackle this disease. And they know they have the patients in mind, and they're often talking about, okay, but what does it really mean in the clinic? I think that's an important question that often a lot of, researchers, they don't mean to, but you can forget that if you don't see it every day. And so that's something that I'm very hopeful for, and I'm really excited for the field to continue to advance.
Thanks, Sage. And thanks again to our friends from Johns Hopkins for a very informative and very hopeful evening. Before we all say goodnight, I really hope that this information was useful to you. You will be receiving a survey along with the link to this recorded presentation. Please make sure that you fill out the survey. We want to do this again. We want to do this more often. We want to hear from you. We want to hear what your questions are. We want to be able to provide webinars that are useful. So fill out the survey, and we'll be doing more of these. And again, I'd like to thank everyone for joining us the evening of World Pancreatic Cancer Awareness Day. Please join us again for another webinar, which we will be live streaming on December 8th. And you'll be hearing from Dr. David Tuvison, our chief scientist, and Dr. Liz Jaffe, our chief medical advisor, about progress and promise in 2022. What have we learned this year and where are we going in 2023, and why we all have more hope than ever before that we will be transforming pancreatic cancer into a curable disease. Thank you all for joining us. Thanks again to our friends at Johns Hopkins, and hopefully we'll see you all again on December 8th. Thank you all.